Thank you for taking some time this afternoon to come hear our story and the story of these other great companies that are up here. ISTO Technologies is what we like to refer to as an advanced staged orthobiologics company. We have three major programs that we're working on, two of which are cell-based, one targeting articular defects in the knee. The second cell-based program is targeting discogenic back pain. We also have a third program, a second platform technology that is a synthetic biomaterial that's used in spinal fusion surgeries. I think everyone can agree that both the spine and the markets that we're targeting in terms of cartilage regeneration are huge opportunities for a company like ours. We do have a strong corporate partner in Zimmer in place for their knee program. The other two programs, we do not have any partners at this point in time. Here's an overview of the three programs that we have. The company started based on some technology that was developed at Barnes Jewish Hospital in St. Louis, and it dealt with the regenerative capacity of juvenile cartilage cells compared to adult cartilage cells. And that was really the basis of the de novo ET program, or what was at the time called neocartilage. And we partnered that program with Zimmer back in the early part of 2002 and have been in development and clinical work since that time. And I'll tell you a little bit more about that program and what makes us unique in terms of this cell therapy program. We are in a phase 3 human trial with this program in partnership with Zimmer, and we're very excited about what this opportunity has. Our second kind of cell-based program, as I mentioned, is a program called Nu-Q. This is a program that we started clinical trials back in 2010, and it's focused on the treatment of discogenic back pain. And by that we mean people that have lower back pain that are non-responders to conservative therapy. And this is a cell therapy designed to prevent them from having to progress to a surgical option. Our third program and our second platform technology is a synthetic biomaterial called NQ. It's a unique entanglement of PLGA and hyaluronic acid, and it's being used in spinal fusion surgeries. We launched this program in 2008 and are generating revenue from it at this point in time. We have a fourth program that's really a subset of the de novo ET program. It's called de novo NT. It's a natural tissue that we work in terms of processing the juvenile cadaveric tissue that we work with. And it's been used in probably 3,000 to 4,000 surgeries to date. It is commercially available through our partnership with Zimmer. It is different from our ET program in terms of it is not manipulated. It's just a true natural tissue that we're processing. We'll go very quickly through this, but as you can see, the programs that we're focused on, both arthritis and back and spine pain, are two of the largest unmet medical needs that are out there in terms of two of the leading causes of disability in the U.S. This is the initial discovery that formed the basis of our cartilage programs, and it deals with like I said, the regenerative capacity of our juvenile cartilage cells. As you can see, and I like to tell my 11-year-old daughter that she's already over the hill now at the age of 11 because her cartilage won't regenerate. Or it tends to lose that capacity very rapidly. We tend to work with the young cadaveric donors in terms of through our relationships with tissue procurement organizations located throughout the U.S. One of the other things that's interesting to note in terms of our work with the cartilage for both our ET and our Nu-Q programs is that cartilage is an immune-privileged tissue. So we have quite a bit of research proving that. And there, as I said, we've treated with our NT program probably 3,000 to 4,000 patients, and through our clinical work for both ET and Nu-Q, we've not had any safety issues with any of those programs in terms of rejection or any other issues with regards to the processing that we do. As I mentioned, as you're probably well aware, when you're dealing with donor tissue, there is a limited supply that you're able to work with. And so a lot of the early work that we focused on dealt with our expansion technology, our ability to take these cells and go through multiple doublings and expand this to really develop a true commercial scale type program. So for both our ET and our Nu-Q program, we have a proprietary process where we go through an expansion process, and then we're able to either divert those cells to our Nu-Q program or take the next step to growing a living cartilage graft. And out of a single donor, we have the capacity at this point in time to grow 3,000 to 5,000 either doses of Nu-Q or 3,000 to 5,000 grafts for our ET program. And this will have a shelf life for the ET program of a couple of months, depending upon testing requirements as we work through the FDA process. And for the Nu-Q program, it'll really be kind of an indefinite life because those cells are actually frozen down and then thawed when they're used. Here's a picture in the upper left-hand corner of what the graft looks like for our ET program. And as I mentioned, this is a program that we're in partnership and conducting the clinical trial with Zimmer. As you can see on the bottom, There's a cartilage defect in this patient's knee. It was done as an open procedure in the phase 1. We are working on, we'll be working on developing this as an arthroscopic procedure going forward. We had a number of the patients in the phase 1 that agreed to second look arthroscopies and biopsies. And as you can see in the picture in the middle, this was something that this graft that was implanted, it was implanted in this 3 to 4 weeks of non-weight bearing, and then a typical recovery that you would have from any type of knee procedure, be it microfracture, shaving, debridement. We had full integration of the graft and beautiful performance of that patient, and they were able to resume their daily activities. More importantly, I think, is that what we were able to get some biopsies of these grafts after a year, and we're able to determine that what we were growing was true hyaline cartilage, not the fibrocartilage that you may see from a microfracture procedure, but truly the own natural cartilage that your own body would regenerate and have if you were at a younger age. Comparing our cartilage technology, I think it's important that when you look at other cartilage technologies that are in development, those are all autologous approaches. One, they're working with adult cells, which, as I showed you earlier, our juvenile cells that we work with are up to 100 times more potent in terms of their ability to regenerate new matrices. Secondly, ours is a one-step procedure. It's not a two-step surgical procedure that requires a great deal of training and difficulty in terms of working with the surgeons. Secondly, our expansion technology offers a scale that cannot be found in those type of autologous approaches where you're having to do kind of that personalized medicine approach. So from a business standpoint, this is a very attractive product for us as we go forward. The Nu-Q program, as I mentioned, we started our clinical trials in 2010, and this is a innovative biologic disc repair. So this is, as I mentioned, we're targeting people that have chronic low back pain, and it's an outpatient procedure. So what you see on the picture on the left is the actual injectable, the needle that's used to inject these cells in an outpatient process. So the market that we're targeting are people that have chronic low back pain. They don't respond to physical therapy. They don't respond to rest. They don't respond to steroid injections. And today, the only options are surgical. So the surgeon will go to them and say, you either need a fusion, a discectomy, a disc replacement, or something that's very invasive. We think there's a real market opportunity here to basically treat these patients on an outpatient basis, inject these cells, and give them a chance to regenerate the disc nucleus and also give the annulus a chance to heal itself. We did start our phase 1. We treated 15 patients. Here's kind of the inclusion-exclusion criteria that we used. All our subjects have completed our 6 and 12-month follow-up. We've been very pleased with the results. We have submitted our interim report to the FDA and expect to hear back very soon in terms of what that next step will be from a clinical program. We're hoping to be able to proceed directly to a phase 3 like we did with our ET program. But when dealing with the FDA, you can never be certain. I am happy to report that for the primary outcome we're looking for is safety in this study, and we have not had any serious or unexpected adverse events at this point in time. And we've seen strong efficacy data on a secondary basis. More importantly, we see continued improvement continued progression from 6 to 12 months, which shows us that the product is working, and we're very excited about what this holds. Changing gears a little bit, this is our commercial program, a program called NQ that's a kind of a unique, as I mentioned, entanglement of PLGA and hyaluronic acid. And we think that's very unique within the spinal fusion world in terms of this is a bone graft substitute and extender. It's not a growth factor. It's not a calcium-based product. It really uses that hyaluronic acid to try to mimic kind of the mechanism of action of some of the growth factors, but in a much safer pattern at a much lower cost point, which is very important for the hospitals that we're dealing with. Our primary market that we're looking at is spinal fusions. This does have applications in the trauma market, sports medicine, and foot and ankle. But at this point in time, the spinal market is the biggest market for these type of products. We are, I guess, very excited about what this product and in this environment, the capital that the cash flow that this provides allows us to continue to do development on our cell-based products. As I mentioned, this product we started it in 2008, and to date we've treated over 12,000 patients, and there's been no safety issues with this product as well. We think that this is also a product that offers the opportunity for for further development in terms of other areas of soft tissue, whether it's osteochondral, whether it's cosmetic, whether it's urinary incontinence. This is a program that we truly view as a platform technology. From a financial overview, we are always glad to be able to talk about our revenue growth that we've had. We did a little over $14 million in 2011. We're hoping to do over $20 million in 2012, and are very excited about what that cash-generating capability offers for us moving forward. The company, like I mentioned, is we feel we're unique somewhat in the orthobiologic space, and that we're very diverse in terms of, have programs in sports medicine, we have programs in spine, we have both cell-based programs, we have biomaterial programs as well. The company's raised about $45 million since inception. Our major shareholders are Alafi Capital, Zimmer, and Mid-America Transplant Services and Ascension Health Ventures. Mid-America is one of the tissue procurement organizations that helped the company get started. Our goals for this year are to really continue to drive our NQ sales going forward, and also look to take the next step with our Nu-Q clinical program, as well as continue to progress through the phase 3 trials with our partner Zimmer for the ET program. We are interested at some point in exploring corporate partnering opportunities for both our Nu-Q and our NQ programs, and eventually position the company for an exit. So that's the ISTO story, and I appreciate your time and attention.